Commensal E. coli strains in birds have the potential to become pathogenic and cause colibacillosis infection. Colibacillosis is caused by a group of extraintestinal pathogenic strains of E. coli, which are known as avian pathogenic E. coli, or APEC. It can cause great economic burden at any stage through the poultry production pathway, and there is no current diagnostic tool to distinguish between commensal and pathogenic E. coli. Our goal was to leverage publicly available microbial genomic data as well as E. coli isolates from avian necropsy cases at the Penn State Animal Diagnostic Lab to identify virulence factors characteristic of APEC strains, which could serve as potential targets for molecular diagnostic test development. We retrieved over 312,204 E. coli genome records from the NCBI GenBank database. Associated metadata from the biosample database was used to filter for genomes derived from poultry with a disease status of either healthy or colibacillosis. The genomes were screened for virulence factor genes using the tool Virulence Finder. Fisher's exact test was then applied to assess their association with colibacillosis, followed by multiple testing correction to adjust p-values for statistical significance. We collected about 450 E. coli cases from necropsy cases at ADL over a 1-year period. We conducted whole genome sequencing of these isolates, and assembled genomes were screened for virulence factors using the same tool. We correlated the virulence genotypes with pathology findings of associated diagnostic cases. The top 12 virulence factors significantly associated with colibacillosis infection, along with the corresponding odds ratios and major roles of the genes, are included in the table. At the top right, this volcano plot shows the virulence genes associated with colibacillosis based on the NCBI biosample metadata. The gene with the highest odds ratio indicate the highest association between the presence of this gene and a colibacillosis. The y-axis represents the statistical significance. Below the table is a heat map of co-occurring virulence factors. The darker red colors on the heat map signify a stronger correlation between the genes, highlighting their co-occurrence. Beneath the heat map, there is a network illustrating the most frequently co-occurring genes, along with those identified as statistically associated with colibacillosis status in NCBI metadata. Finally, pathology findings were correlated with the presence of various detected virulence genes. Polyserositis was the most common condition in cases where E. coli was identified as the primary pathogen. In the bubble plot, each diagnosis on the y-axis is paired with virulence genes on the x-axis, with the bubble size representing the proportion of samples containing each gene. To conclude, major virulence factors associated with APEC strains were identified. These results can contribute to future diagnostic tools and control of colibacillosis through designing an improved molecular diagnostic test for the identification of APEC strains, as well as establishing criteria for the selection of strains for autogenous vaccines. PCR can specifically target the most important genes, such as FYUA and IRP2. We would like to acknowledge the Center for Poultry and Livestock Excellence, who funded this study, as well as the staff at the bacteriology and pathology sections of the Penn State Animal Diagnostic Lab. Thank you.